Hello and welcome to my channel. Now, before we begin with the topic of protein biology, let's briefly discuss what is biochemistry. Biochemistry is the study of chemical processes within living organisms, and it involves a lot of chemistry. We can broadly categorize the field of study on the basis of functions performed by biomolecules inside a cell. These biomolecules either perform the function of information, structure, and fuel, and include DNA and protein, or they do not provide information but are important for the structure and fuel components, including carbohydrates and lipids. Each one of these biomolecules is made up of monomeric subunits, which are called nucleotides in DNA, amino acids in protein, monosaccharides in carbohydrates, and fatty acids in lipids. Coming back to the topic of today's video, that is protein biology. Proteins, as we know, are important for a living system as they manage information, structure, and fuel, and are important for various functions of a cell. Before we proceed, we need to know two important definitions related to proteins. First of all, what is a protein? A protein is a nitrogenous biomolecule made up of long chains of amino acids serving essential functions in a living organism. And what is an amino acid? Amino acids are organic compounds containing a carboxyl group, an amino group, a side chain, and a central carbon atom. These molecules are chiral or asymmetric molecule, which is a very important property of amino acids, as we shall discuss in the later slides. Represented here is an L-amino acid, where the central carbon is called C-alpha. Attached to the C-alpha are the amino group, the carboxyl, and the side chain. We can classify amino acids based on various properties, but here we will stick to the classification based on polarity of side chain. On the basis of polarity, which means distribution of electrons such that it allows the amino acid to interact with water molecules. We can classify the amino acids into three types, which are non-polar, which cannot interact with water, and these include glycine, alanine, valine, leucine, isoleucine, proline, methionine, tryptophan, and phenylalanine, as represented here by their three-letter code. Second, we have polar uncharged amino acids. These include serine and threonine, which have a hydroxyl group in their side chain, tyrosine, which has a phenol group in its side chain, cysteine, which has a SH group, and asparagine and glutamine. Whereas the third category is polar charged amino acids, which are divided into two types: positively charged amino acids, and these include lysine, arginine, and histidine, and negatively charged amino acids, which include aspartic acid and glutamic acid. Here we have a representation of non-polar amino acid, where we can see that in the side chain we have a methyl group, therefore it is non-polar. Then a polar uncharged amino acid, which has a hydroxyl group in the side chain, and polar charged amino acid, which has a positive charge in the side chain. Now let's discuss about the properties of amino acids. First of all, their configuration. So what is configuration? Configuration means arrangement of atoms in a molecule. And for amino acids, we have various systems to define configuration. The most common system of configuration we use is the D and L system. D and L system is a nomenclature based on the direction of rotation of the plane polarized light, and this rotation is measured when we pass a monochromatic light through an amino acid suspension in a polarimeter. We will not be discussing the details of how the polarimeter works and how we calculate optical activity in this video. However, let's discuss the DL system in detail. So in amino acids, we know that the L form is the carbon form. Here is a representation of L-alanine, where L means levorotatory, that is, it rotates the plane of polarized light in anti-clockwise direction. As you can see here in the diagram, that the arrow represents rotation of light in the anti-clockwise direction, and L form is the common form in case of amino acids. Also, an important point to be noted here is that the amino group in L form is always on the left hand side. Second, we have the D form, D-alanine, which is dextrorotatory, which means that the D-alanine rotates plane of polarized light in the clockwise direction as represented by the arrow. And these forms are rare in nature and are usually found in the cell wall of bacteria and in the peptide antibiotics. Another system used to decide configuration of amino acids is the R&S system, which is a nomenclature based on the atomic number of elements around the chiral carbon center. Let's look at an example to understand this. Here we have L-alanine, which is also called as S-alanine or the sinister form. So how is this decided? Here we can see that the nitrogen present in the amino group has the highest atomic number, so we assign the number 1 to the amino group. Then we have carbon on both the side chain as well as the carboxyl group. So in cases where you encounter that a same atom is attached to the central carbon, then we have to look for the second and subsequent atoms. So in the case of L-alanine, the second number will be assigned to the carboxyl group because the atomic number of oxygen is more compared to the atomic number of hydrogen. And finally, number 3 is assigned to the methyl group in side chain. Therefore, if we draw an arrow to see the rotation, here we see that the arrow follows from amino to carboxyl and then to the methyl group, which is anti-clockwise, and that is why L-alanine is S form or sinister form. Similarly, D-alanine will be R form or rectus because here the order gets reversed. Therefore, the arrow which we mark to represent the atomic numbers will be in the clockwise direction. Now, except for one amino acid, all the amino acids which are L form are always sinister, whereas the amino acid whose L form is not sinister is L-cysteine. L-cysteine exists as R form because of the presence of sulfur in its side chain. Here we can see that we assign number 1 to the nitrogen of amino group, but we do not assign number 2 to the carboxyl group because in the side chain of cysteine we have sulfur attached to the carbon of the side chain, due to which the atomic number of the side chain becomes greater as compared to the carboxyl group, and hence the arrow represented here will be in the clockwise direction, and that is why L-cysteine is also an R form instead of being S form. Now let's talk a little about non-standard amino acids. These are amino acids apart from the 20 standard amino acids which we discussed in the classification of amino acids. Non-standard amino acids are modified chemically by post-translational modifications or PTMs, and the most common modifications observed in amino acids are hydroxylation, phosphorylation, and carboxylation. The figure here represents how post-translational modification happens. First of all, we have a gene in the DNA which gets transcribed into RNA, which later on gets spliced to become mature RNA, and this RNA undergoes translation to produce amino acid chain or polypeptide. But after the polypeptide has been formed, it undergoes further modifications at several amino acid residues to finally give rise to the protein in the cell. Now, what is the significance of non-standard amino acids? First, in collagen, which is a structural protein, we have 4-hydroxyproline and 5-hydroxylysine, which are hydroxy forms of proline and lysine. Then in prothrombin, which is important in blood clotting, we have gamma carboxyglutamic acid, which helps in calcium binding. Then 3 amino acids which are very common during the signaling net pathways are serine, threonine, and tyrosine, which gets phosphorylated. Also, in glycoproteins, we have amino acids which are modified by attachment of sugars to the nitrogen and to the oxygen atoms. Then we also have a chemically synthesized amino acid called selenocysteine, which has selenium in place of sulfur in the cysteine. And finally, these amino acids also find medicinal use. For example, L-DOPA is used for the treatment of Parkinson's disease. So the final topic of this video is titration of amino acids. In chemistry, titration is a commonly used quantitative technique to determine the concentration of solutes dissolved in a solution. In case of amino acids, we use titration curves to identify the concentration of amino acids as well as their acid dissociation constant or the pKa value. Now, amino acids are polyprotic acids, which mean they can lose more than one proton or H plus ion, and they exist as zwitterions or dipolar ions in solution. That is, in a solution, an amino acid has both a positive as well as a negative charge. The amino and carboxyl group of amino acids are ionizable, which means they can produce ions, and additional ionizable groups are also present in the side chain of the amino acid, and these can also be titrated. So the curve which we obtain during titration of amino acid depends not only on the amino and carboxyl group but also on the side chain. The pK of an amino acid is the pH at which the protonated form which is represented as HA, and unprotonated form, that is A minus, forms of an ionizable species are equal, or it is the pH at which solution resists change in pH effectively, which means even upon addition of more acid or base to the solution, the pH will not change when the pK has been achieved. pK can be calculated using Henderson-Hasselbalch equation. In this video, I am not going to discuss the derivation of this equation, but for an amino acid solution, when the protonated form gets dissociated into the ionic forms, this equation is represented as pH equals pK plus log of the unprotonated form, that is concentration of A minus divided by concentration of the protonated form, or HA. So a titration curve is a plot of pH versus volume of titrant or mole equivalence added to a sample, and if we use a pH meter, then upon addition of acid or base to the amino acid solution, we can calculate changes in the pH and thereby calculate pKa of the amino acid and its amino acid concentration. Say for example, we use a base as a titrant, then initially we observe that the predominant species of ions present in the amino acid solution will be cationic form, which upon increasing the pH will become a zwitterion, and finally we see that the predominant species will be the anionic form. This is represented here by this graph, where on the x-axis we have volume of sodium hydroxide or the titrant, and on the y-axis we have pH. So we see that as we increase the volume of sodium hydroxide, the pH also increases, and we see two inflection points on this graph where we observe pK1 and pK2. In between, we see that there are equivalence points in these plots. These are the points where the positive and the negative charges are equal, and is important to determine the property called as isoelectric point. So what is isoelectric point? Isoelectric point, or Pi, is the pH at which the amino acid has net zero charge, and this is what we see in the graph in the first equivalence point. And isoelectric point usually exists in between the two pK values. That is why in equation isoelectric point is always an average of the pK1 and pK2. Isoelectric point is important for the study of amino acids because this is used for the separation of amino acids, or is important in what we refer to as the 2D page analysis, which we will discuss in later videos. Thank you for watching this video. Like, comment, and subscribe if you like it. Stay tuned for more such